I'm going to talk about mitochondrial adaptation and metabolic flexibility in drug resistance of acute myeloid leukemia. I will do first a short introduction about mitochondrial function, and then I will present indeed, I will present you an overview of our major results we got since I came to the Cancer Research Center of Toulouse 10 years ago, living from Philadelphia, from UPenn. And I will try to convince you about our current working model of the energetic basis of drug resistance in acute myeloid leukemia. So, as you know, since Otto Warburg and his famous aerobic glycolysis, changes in not only in glycolysis but also in other metabolic pathway are hallmark of the cancer biology. Originally, Otto Warburg thought that this metabolic phenotype of cancer cell was in fact the consequence of deficiency, alteration in a mitochondrial function. But now we know perfectly that mitochondria, in fact, in cancer cell are perfectly active, just different from normal cell. So mitochondria are the site of the ATP production through the oxidative phosphorylation, consisting on the respiratory change with a 4 redox enzyme complex from complex 1 to complex 4. And that respiratory change is coupled to ATP synthase complex 5, responsible for phosphorylation of ADP into ATP using the proton gradient generating by complex 1, 3, and 4. So to support this OxPHOS activity, TCA cycle and fatty acid oxidation, generating electron donor from NADH for complex 1 or FADH2 for complex 2. So as shown in this schema, major respiratory substrates such as glucose, glutamine, and lipid are maintaining this fatty acid oxidation and TCA cycle activity to support energy production. In addition to be the site of the TCA cycle, Krebs cycle, and the mitochondrial respiration, mitochondria are the hub of metabolism. And in fact, all metabolic pathways in cell are at least for one biochemical step dependent on mitochondria. And one of the best examples we have in cancer and in leukemia, it's the pyrimidine biosynthesis, where the critical step is the DHODH, dehydroorotate dehydrogenase step, using electron transfer change and depending on the OxPHOS activity to produce or rotate and to support the pyrimidine biosynthesis. In addition of that, mitochondria are also implicated in calcium homeostasis and signaling in redox balance, being the major site of ROS production. All of those essential functions are regulating the cellular bioenergetic balance. And finally, mitochondria are playing a crucial role in programmed cell death through the mitochondrial intrinsic apoptotic pathway. And as you can see in this beautiful schema, there is a very tiny relationship between the energetic OxPHOS activity, the ROS production and generation, and the regulation of the apoptosis through the mitochondrial permeabilization transition pore that release apoptotic signals such as cytochrome C. Drug resistance and minimal residual disease are the major therapeutic barrier for the treatment of acute myeloid leukemia, and it's quite critical to identify the mechanism of resistance in relapse initiating drug tolerant cell. And For the purpose of this talk, I will use the abbreviation RIX for this relapse initiating resistant cell. And initially, we ask the question whether mitochondrial function might be essential to this RIX. So using a PDX model, we first identify that RIX display high OxPHOS gene signature and an hyperactivation of the mitochondria OxPHOS activity through an increase in mitochondrial membrane potential, mitochondrial respiration, ATP production, an increase in ROS content, and also an increase in fatty acid oxidation. This hyperactivation of the mitochondria metabolism in RIX is in fact the consequence of an increase in mitochondrial machinery, not only the mitochondrial biogenesis, but also the mitochondrial transfer from stromal cell, bone marrow stromal cell, to AML blasts. So currently, we are also demonstrated that mitochondria iron metabolism, heme bio pathway, and ROS defense mechanism are strongly related and connected to this high OxPHOS mitochondria. And associated to this special metabolic phenotype, we are observing right now an increase in BCL2 dependency through the translocation of BCL2 to the mitochondrial membrane in this cell. And very recently, we published a Nature Communication paper showing that increase in BCL2 with an increase in VDAC, in fact, increase the mitochondrial ER contact site that support mitochondrial calcium contact through the influx of ER calcium to the mitochondria. And this calcium will stimulate not only pyruvate dehydrogenase, but also TCA cycle dehydrogenase activity that support OxPHOS. But in addition of being playing a crucial role in a calcium influx, MERCs are also one of the major sites of the autophagosome formation. And in this paper published last month, we are showing that increase in fact, we have an increase in lipophagy, in autophagy of lipid that release free fatty acid to support fatty acid oxidation. So we are demonstrating, and we are also convinced that mitochondria, in fact, auto-regulate their own substrate availability to support OxPHOS in this resistant MR cell. So based and consistent with this high OxPHOS status, RIGs are more sensitive to mitochondria inhibitor. And in fact, if you target any aspect of the high OxPHOS, it can be directly at the electron transfer change, but also other kind of function that regulate OxPHOS, you will sensitize resistant cell to RNC. And also this summer, we were quite happy and lucky to publish a second story in a Cancer Discovery, where we showed that high OxPHOS phenotype, in fact, reflect a mitochondrial adaptation induced by a specific transcriptional pathway upon cytarabine treatment. And in fact, cytarabine will lead to an increase in CMP and ROS that activate protein kinase A signaling pathway and ATF4 that support mitochondrial stress response, leading to the upregulation of PGC1 alpha. This transcriptional factor, in fact, one of the major regulators of the fatty acid oxidation and mitochondrial biogenesis. And we are currently showing, in fact, this mitochondrial stress response is induced by a transient mitochondrial dysfunction induced at the early stage of the drug application through the transient decrease of the mitochondrial membrane potential, leading to an increase in mitochondrial DNA release that supports this induction of the pathway and the mitochondrial adaptation, priming the ML cell to resist to drugs. So since this first paper we published in Cancer Discovery, there is a plenty of group and with a beautiful publication, as you can see here, showing that changes in mitochondrial energetics, metabolism, or structure are hallmarks of drug resistance. It can be chemotherapies, also other chemotherapies, but also targeting therapies used in this disease, such as ADH mutant inhibitor, FLT3 inhibitor, or BCL2 inhibitor. And all of those work are suggesting to us that OxPHOS flexibility and mitochondrial adaptation are in fact pleiotropic non-genetic mechanism driving AML heterogeneity and drug resistance. As I told you in a couple of slides ago, we observe with this high OxPHOS phenotype an increase in BCL2 relocation to the membrane, suggesting inhibiting BCL2 with venetoclax in association with cytarabine might be very relevant therapeutic approach. And as you know, there is a strong therapeutic revolution with venetoclax in AML, and recent clinical trials are showing venetoclax plus low-dose Arac are better than low-dose Arac alone in unfit patients. So we have demonstrated in our PDX model that combining these two drugs, venetoclax plus Arac, we observe a significant synergy and better effect than Arac alone or venetoclax alone. So we decided a couple of years ago, I think 1 year ago, last summer in fact, to try to identify the mechanism of resistance in this duplet therapy compared to the single agent from one PDX after establishing the disease and treating with single agent in blue, in gray with venetoclax alone before treatment, and then the duplet therapy in orange. So we use single cell RNA-seq to try to identify a therapeutic resistant marker and mechanism. And as you can see here in this T-SNE map, we observe a very strong transcriptional heterogeneity, not only before treatment, but also after single agent, venetoclax and Arac. And they are perfectly this heterogeneity, it's exactly the same in these three conditions. But when we look at the heterogeneity of the subclone after the duplet therapy in orange, we were quite surprised. We identify 3 de novo emerging transcriptional subgroup after these duplet therapies in vivo in PDX. So using gene signature published by Peter Van Galen in this famous Cell paper a couple of years ago, we were able to show that this transcriptional heterogeneity overlap with a phenotypical heterogeneity. And for example, cluster 13, it's enriched in gene-related to HSCs and progenitor gene signature, while complex 6, cluster 6, it's mostly a pro-monocytic cell. So we also use a couple of algorithms to try to better understand the mechanism of resistance, and using CENIC in collaboration with our collaborator Christophe Marine and Aurélie Boussa in Brussels, we were able to show this 2 big cluster 13 and 6 in fact was enriched with MITF TP53 regulon that specifically activate in residual disease after the duplet therapy. And what is very interesting, these 2 regulons are known to be a key regulator of the mitochondria oxidative phosphorylation in other type of cancer, such as melanoma, for example. And we decide to look at the upregulated genes in this tree-specific and tree de novo emerging cell cluster compared to the genes in other cell cluster. As you can see in this Venn diagram, there is only 9 genes in common with these 3 cluster. But surprisingly, among these 9 genes, 3 was related to the assembly and the chaperoning of the electron transfer change complex 1. So we decided to use an OxPHOS inhibitor, an ETCI inhibitor, Ajax, in collaboration with Marina Konopleva and Joseph Mazarek at the MD Anderson in Houston, and to try to target this minimal residual disease after 1 week of treatment, and we treat in consolidation phase for 9 weeks with this low dose ETCI inhibitor. And we follow up the relapse in bone marrow, in peripheral blood and bone marrow environment. And as you can see here, why RC alone and venetoclax plus RC alone relapse after 7 to 8 weeks, even if after 14 weeks, we still not observe any significant relapse in the cohort with the triplet therapies, even if 4 weeks after the stop of the administration of the Ajax. And that reflects a significant reduction, 2-log reduction, 4 weeks after the end of the treatment compared to a single agent, as you can see here in total cell tumor burden. So again, we are demonstrated with this novel venetoclax combo that OxPHOS, ETCI flexibility and mitochondrial adaptation, it's a crucial mechanism of resistance to drug in AML. So based on our study, we observe with all drug we use in our study, we observe the activation of 3 different transcriptional factor regulatory networks, but we have an overlap on PGC1 alpha in these 3 different networks, suggesting and confirming the central role of PGC1 alpha in this response to this different treatment. So now we are wondering if blocking the transition to mitochondrial adaptation and blocking the transition between mitochondrial stress response to mitochondrial adaptation will be maybe a promising approach. And in fact, we have a proof of concept study using anthracycline as an inhibitor of PGC1 alpha. And we are currently revisiting the mechanism of synergy with RC, proposing this mechanism in addition to the regular DNA synthesis inhibition with these 2 chemotherapies. So based on this work, we are currently proposing this model, this biphasic mitohormetic model of drug resistance, where at the early stage, drug induce transient mitochondrial dysfunction leading to mitochondrial stress response and mitochondrial adaptation at the end of the, or as a later stage. That mitochondrial adaptation, in fact, will support the metabolic and energetic flexibility that prime AML cell to be resistant. So our current question is to know what metabolic pathway can support this high OxPHOS activity and mitochondrial phenotype in RIX in vivo. As I told you from our initial paper in 2017, we observe an increase in fatty acid oxidation and an increase in CD36 uptake of lipid or extracellular lipid to support the mitochondrial OxPHOS. But several other study and very recent study from David Scadden lab are showing very elegantly that aspartate, extracellular aspartate, derived from stromal glutamine might support pyrimidine. And as I told you in the introduction, to have this pyrimidine biosynthesis activation, you need to have DHODH and the mitochondria OxPHOS activity. And another very beautiful paper from Craig Jordan lab showing that cysteine might be a key amino acid to support glutathione and support the mitochondrial oxidative phosphorylation by activating succinate dehydrogenase through the glutathinylation of succinate dehydrogenase. And in our cancer discovery paper this summer, we showed that extracellular ADP and ATP, in fact, are regulating mitochondrial OxPHOS through the activation of CD39, P2RY13, and the activation of PGC1 alpha that lead to an increase in fatty acid oxidation and mitochondria adaptation. So based on all of that, we are proposing this metabolic model of drug resistance, where at the beginning of the treatment of the drug administration, you will have a metabolic collapse, a transient metabolic stress that lead to a metabolic selective pressure that supports the emergence of RIX and that support also the emergence of this high OxPHOS phenotype, leading to the metabolic adaptation of those cells that support first the drug tolerance and then subsequently to the drug resistance that initiate the relapse. And we are thinking this model in AML might be quite relevant for multiple therapeutic resistant cancer type. So there is still many unaddressed question about high OxPHOS or OxPHOS RIX. Here it's three we are currently thinking about. Does energetic flexibility drive metabolic heterogeneity of resistant cell population in AML? We were wondering how host metabolism and diet might support this OxPHOS metabolism of RIX. And finally, we are trying to understand whether host and cell autonomous autophagy might be a key regulator of this hyperactivation of mitochondrial metabolism in RIX. So I want to acknowledge all the people who did the work in my team and the former member, the clinical part, because we are using a lot of PDX and we need clinicians to be involved in our story, and we also our animal facility and our collaborator and founder. And thank you so much for your attention.